Good morning, class. So before we start our lesson, may I ask Miss Marcos to please lead us into prayer? Amen. So before you take down your seat, please pick up pieces of papers and arrange your chairs properly. Okay, you may sit down. So let me check your attendance first. Is anyone absent? Okay, good. So all of you are present. So before we move on our next topic, can you recall what our last topic was, Miss Olida? You're right. We tackled about meiosis, which is a cell division in which four haploid cells are formed from a single diploid cell, which usually occurs in the reproductive organ or gonads of an organism. Can someone also briefly define the process in meiosis 1, which includes the phases of prophase 1, metaphase 1, anaphase 1, and telophase 1 with cytokinesis? Yes, Miss Moreno, you're right. How about the process in meiosis 2? Yes, Miss Robredo. Yes, you're right. Meiosis is very important in maintaining the number of chromosomes in an organism, and it is also important in heredity. There are traits that we inherit from our parents, for example, our skin color, the color of our eye, and many more. So I will be giving you this activity, and you're going to check the appropriate boxes. Identify the following traits if they belong to your mother, your father, your brother, your sister, or to you. If you are the only child in your family, do not check the boxes below the brother and sister, and please make sure to check what is appropriate only for you. So please do this for 5 minutes. Please pass this. Okay, 5 minutes already done. So based from your observations of the different traits of your family members, who do you think looks most similar to you? Yes, Miss Cabanag. Oh, how about you, Miss Ebrio? Okay, and how about you, Miss Tamiray? Yes, okay. So how about your family members? Who looks most different from you? Miss Laksan? Okay, how about you, Mr. Arsley? Okay. So always remember that every individual is unique because everyone has a unique set of traits. So those traits that we inherit, that we get from our parents, are called inherited traits. Our topic for today is about Mendelian genetics. There are different forms of living things around us. There's a lot that we cannot name them one by one. In this image, what do you think are the similarities and differences among these organisms? Yes, Miss Jokno? How about you, Miss Gadon? Yes, diverse forms exist among living with similar species or among different species. Our learning objectives for today is first, to discuss Mendelian genetics. Second, is to solve and predict Mendelian phenotypic and genotypic expressions of traits in a monohybrid cross. Third, to solve and predict Mendelian expressions of phenotypic and genotypic expressions of traits in a dihybrid cross. And fourth is to predict phenotypic expressions of traits following simple patterns of inheritance. Please take note that the knowledge you will learn in this topic will help you understand why such traits are inherited by such organisms. Gregor Mendel is the father of genetics. He is also an Austrian monk who gives a better understanding of heredity and variation by studying the characteristics of garden peas or Pisum sativum. So why did he choose garden peas instead of other plants? He chose garden peas because, first, it is self-pollinating. Second, it is fast-growing. Third, it is easy to raise. Fourth, it can produce a lot of products in a little span of time. And lastly, it has different traits that are noticeable. On his study of garden peas, a new branch of science was discovered, and it was called genetics. So, what is genetics? Genetics is the study of heredity and the factors that affect the transmission of genes to determine inherited characteristics from one generation to another. While on the other hand, heredity refers to the traits that are transferred from the parents to the offspring. So in this image, we can see the 7 contrasting traits that Mendel had studied in green peas. So we can see the different traits according to their seed form, the seed colors, the pod form, the pod color, the flower position, the seed coat color, and also the stem length. The pure breeding peas were called the parental or P1 generation. The parental or the P1 generation is the first set of parents cross in which their genotype is the basis for predicting the genotype of their offspring. When cross-pollinated, the resulting offspring is called the first filial generation. The first filial generation is comprised of offspring resulting from a cross between the individuals from parental generation. It is also called hybrids because it resulted from a cross between two purebred plants with different traits. There are 2 types of crosses that may occur among organisms. The first one is monohybrid cross. This is a cross using only one trait. From this image, we can see that a green pod is crossed with a yellow pod. The trait that is crossed is the pod color of the garden pea. Take note that only one trait is crossed in a monohybrid cross. When 2 first filial generation is crossed together, the offspring is called the second filial generation or the F2 generation. From this example, we can see that the parental is capital G, and small letter g, small letter g. Thus, the F1 is capital letter G and small letter g. Now, to determine the F2 or the second filial generation, we have to cross F1 with F1. We have to cross heterozygous G with heterozygous G. Thus, the F2 is 3/4 heterozygous G and 1/4 recessive trait of G. Before we discuss the Mendelian principle of heredity, we have to know that Mendel's result of his experiment made him hypothesize that there was a factor in the plants which controlled the appearance of a trait which he called the heredity factor. Further, he also hypothesized that the traits were regulated by a pair of factors called the alleles. The alleles refers to a variant form of a gene, and it is represented by a letter name, either a capital letter or a small letter. The first Mendelian principle of heredity is the law of dominance. It states that in every organism, there is a pair of factors or genes which control the appearance of a particular trait. One pair of alleles may hide or prevent the appearance of the other. The one allele is dominant while the other allele is a recessive trait. The dominant trait is evident and is being manifested by an individual, while the recessive trait is not expressed and is not seen by an individual. Remember that the dominant trait is represented by a capital letter, while the recessive trait is represented by a small letter. For example, we have here R is for round seed, and small letter r, this is the recessive trait, which is for wrinkled seed. The capital letter R is the dominant trait. We also have here letter G, which represent for green pod. G is the dominant, the capital letter G is the dominant, and the recessive one is the G, small letter g, which is the recessive trait. Since genes in our pair, we can identify them either as homozygous or heterozygous. Homozygous are traits which have identical alleles, while heterozygous are traits which have different alleles. For example, we have this capital letter R and small letter r, which means it is homozygous for round seed. And if we have this capital letter R and small letter r, this is heterozygous for round seeds. we can see the difference of homozygous and heterozygous on their alleles. The pair of alleles that is the genetic makeup for a particular trait of an organism is called the genotype, while the phenotype is the observable trait or the visible trait of an organism. From this example, we can determine the genotype and the phenotype of the organism. For this example, we have the homozygous R. It is the genotype, and the phenotype is the round seed. And also we have the heterozygous G, which is the genotype, and then the phenotype is green seed. The second Mendelian principle of heredity is the law of segregation. It states that all pairs of genes for all the traits of an organism segregate or separate from each other during meiosis or gametes formation. Mendel argued that an organism must inherit one factor from the sperm and one factor from the egg. Thus, a new organism receives one factor for each trait from each parent. The third Mendelian principle of heredity is the law of independent assortment. This law states that the distribution of assortment of one pair of genes is independent of the distribution of the one pair. This law applies to genes that are found on separate chromosomes. Thus, one pair of genes is not affected by the other pair. So that is the three principles of heredity according to Mendelian genetics. First, we have the law of dominance, the law of segregation, and then the law of independent assortment. The another type of cross is the dihybrid cross. This is a cross that involves two pairs of genes or alleles, which means there are two traits that are crossed in this dihybrid cross. For example, we have here the homozygous RY crossed with a recessive RY. We have here two traits, the round contrast with the wrinkled, and the yellow seed in contrast with the green seed. An easy way to determine the cross between a monohybrid or a dihybrid cross is through the Punnett square. This Punnett square is devised by Reginald C. Punnett. This is a simple way to determine the possible combination of genes in a given cross. It can help you predict easily the outcome of a given cross. In this, you can determine the phenotype or the genotype of an organism. There are four simple ways or steps on how to create the Punnett square. First, we have to identify the genotypes of the parents involved in the cross and label us as P1 or the parentals. Second, we have to segregate the gametes by encircling them and label them as G. And the third is we have to create a Punnett square and transfer the gametes into their designated area. We have to divide the boxes into 4 for monohybrid cross and 16 boxes for the hybrid cross. And the last one, we can identify the possible genotypes and phenotypes of the offspring. We're going to expand these 4 steps of creating Punnett square by these given examples. Our first example is on monohybrid cross. So as we discussed earlier, we're going to determine the parents first. The parents is heterozygous A crossed with a recessive A. After determining the parents, we are going to determine the gametes. On the first parent, the gametes is A, capital letter A, and small letter a, while the other has the gametes of small letter A and small letter a. After determining the gametes, we are going to place them into their designated areas into the Punnett square. So, we are going to cross capital letter A with small letter a, which the offspring will become heterozygous A, and capital letter A and small letter a, the offspring is heterozygous A. We can also cross small letter a with small letter a, the outcome is the recessive A, and also cross small letter a with small letter a, and the outcome is also recessive A. After the crossing, we can now determine the genotypes and the phenotype of the organism. The genotype of this cross is 2/4 A, A, 2/4 heterozygous A, and 2/4 recessive A. Thus, the phenotype is 2/4 or 50% axial and 2/4 or 50% terminal. So our next example is on dihybrid cross. So as usual, we have to determine first the parent. So we have to cross here heterozygous RY with a heterozygous RY. After determining the parents, we are going to determine the gametes. So the gametes are capital R, Y, capital R, small y, small r, capital Y, and small r and small y. This gametes of the one parent is also the same with the other parent. So as usual, we have to place these gametes into the designated area. We have to, and after that one, we have to cross them to determine the offspring. So here is the Punnett square, and here are the genotypes of this cross. So after determining the genotypes, we can now determine the phenotype of this cross. So as you can see, from these phenotypes, there are traits that are similar with each other. So we have to put them together. So our final phenotype is 9/16 round yellow seed, 3/16 round green seed, 3/16 wrinkled yellow seed, and 1/16 wrinkled green seed. So this is how we create a Punnett square depending on the cross, if it is a monohybrid cross or a dihybrid cross. We have to remember that every time we determine the genotypic or the phenotypic of an organism, it depends on what is asked. If you can put it on percentage or in fraction. It depends on what is asked in the problem. Okay, class, please prepare 1/2 crosswise for our quiz. Okay, that's all. Please pass your papers in the center aisles in the count of 5, 4, 3, 2, and 1. Okay, that's all. Goodbye, class, and see you next meeting.